So in this lecture we will discuss the cultivation of microorganisms, that is bacterial cells. Cultivation, so as you know what is cultivation? Cultivation of crops. Farmers, they cultivate the crops, and then from those crops we get the grains, food grains. So here the cultivation of microorganisms, it involves in vitro cultivation. So we use artificial culture medium. So microorganisms, they can be grown over those artificial culture medium by providing adequate nourishment or adequate nutrition to the bacterial cells, and by maintaining the cells under optimal growth conditions. That is, we have to provide optimum light, oxygen, suitable pH, optimum temperature, and then only it is possible to cultivate the microorganisms. So the key for successful cultivation, we have to maintain appropriate biochemical environment. That is, we have to maintain adequate nutrition, optimum pH, adequate moisture, as well as we have to maintain appropriate biophysical environment. So here the physical factors, that is light, temperature, these are needed to be maintained optimal. So optimum temperature, optimum light, optimum oxygen tension, and adequate moisture, these are important things which are needed for the proper or successful cultivation of any microorganism. That also includes bacterial cells. Next, we have the nutritional requirements of bacterial cells and other microorganisms. So if the cells are phototrophic or chemotrophic, based upon that we have to provide them with a energy source. So for the cultivation, the cells, they require a source of energy. Okay. Phototropic cells, they require a source of light for energy requirement. Similarly, the cells, they cannot grow without the source of electron. So even if the cells are autotrophic or they are chemotrophic, they require an external electron source, and based on the type of electron source, they can be classified as a lithotrophic and organotrophic. If the cells, for example cells like Pseudomonas pseudoflava, this particular variety, or and some other cells, they derive the electrons from the reduced inorganic compounds like lithotroph. They are lithotrophic. The name itself reveals the meaning of the term organotrophic. Organotrophs or organotrophic bacteria such as E. coli, they derive the electrons from organic compounds. Now, additional to these two things, that is the source of energy and source of electrons, the cells, they also require various elements, key elements. For example, the compounds which provide carbon, nitrogen, sulfur, and phosphorus. So supplements of these four vital elements, carbon, nitrogen, sulfur, and phosphorus, they are included in the culture medium. And why these cell components are required? Because these are the major cell components which are present in other cell components, in the proteins, in the phospholipids, in the nucleic acid, as well as in amino acids. For example, phosphorus, it is a major component of bacterial cell membrane. The phosphorus, it is present as a key component in membrane phospholipids, and that is why for the synthesis of phospholipids the cells, they require a phosphorus source. Amino acids, there are various amino acids like tryptophan, methionine. So these sulfur containing amino acids, trypsin, methionine, tryptophan, these amino acids, they can only be synthesized if the cells if they have sulfur as a supplement in their culture media. Similarly, for the synthesis of nucleic acid or DNA, the nitrogen base pairs are required. Sources of nitrogen must be included in the medium. And as all cell components basically they are made up of carbon, we have to provide the carbon supplements in the medium so that we can grow the microorganisms or bacterial cells adequately. Addition to that, the cells also require various mineral salts. They require anions as well as cations. Anions are like phosphate, sulfate, cations like sodium, potassium, some divalent cations like magnesium, ferrous, calcium, zinc. Cells also require the growth factors or vitamins. Those are called as micronutrients, and these micronutrients they are required in very trace amount. For example, vitamin B1, vitamin B6, vitamin B12, vitamin K, hematin, alanine. So if these vitamins if they are not provided, then the cell growth will be very slow, as the cell will take more time to synthesize these vitamins and then they will grow. If you provide them externally in your culture medium, then those vitamins can be directly utilized by the cells and that will help in the direct growth of cells. So these vitamins they are simply called growth factors. Okay. So without these vitamins, without these amino acids, the cell growth will be retarded or cell growth will be very slow. Sometimes the cell growth will not take place. So this happens in case of some typical bacterial cells which require those specific vitamins in the culture medium. Until and unless you provide those vitamins, the cells will not grow. And the main component which is required for the bacterial cell growth or any microbial cell growth is the water. So as you know, 80 to 90% of cell mass is the cellular water. If we measure the cell weight, 90% of it is mainly the cellular water. And that is the reason that we must provide the water as a vehicle in a composition of a culture medium. So water is used as a vehicle and all these other things they are added to the water and then the composition is made ready in which we cultivate the microorganisms. So this is about the nutritional requirements of microorganisms. Cells they require source of energy, source of electrons, source of carbon, nitrogen, sulfur and phosphorus, various anions and cations, the growth factors or trace elements or micronutrients and water.